whether you see the screen or not. Okay, perfect. Okay. Okay, so lovely. Thanks a lot for the introduction. And it's 9 in Sweden, in Uppsala. It's a beautiful morning. I hope you also had a beautiful start of the day in Tehran or any other place that you are listening to this meeting. I would like first to thank the organizer of this meeting, Dr. Mola, and especially Dr. Banabazi and his team behind the Skyroom. And so today I picked a quite broad title, Biodiversity Research in Omics Era. And I thought maybe it can be like an introduction to upcoming lectures after this. So before going to the topic, I just would like to say a few words about the place that I'm working. As I'm working at NBIS, National Bioinformatics Infrastructure Sweden, which is one of the platforms of SciLife Lab. And SciLife Lab is a national infrastructure located in 6 different nodes, different universities in Sweden. And the aim of this infrastructure is to provide service to researchers in different Swedish universities. And in our platform, we do only bioinformatics, and Bengt Persson is the head of this facility. Now let's talk about biodiversity. As you could hear in the opening ceremony, we have one of the main challenges of 21st century. It's COVID-19 pandemic, and it has affected our life to a very large extent. Some of us have lost beloved ones, and until we find a treatment or a vaccine, we have to deal with this situation. But thanks to technology, we are able to be in touch and still interact with each other. It's this particular case in science, and this event wouldn't be possible with these technologies. And we are talking about the COVID-19 as a big problem. Yet there are other consequences of this problem, which one of them will be upcoming economic crisis. And we don't know for how long it will affect our life. But there are even bigger problems, and it is climate change. I know some people do not believe in it, but whether we like it or not, it's a reality. It's not as tangible as the other problems that I highlighted earlier, but it is a true threat to our life. The reason is that on Earth, all the life forms, all the species are interacting, and we are part of a really big system. And it's not like that we are sitting on top of the pyramid. No, we are part or a member of this system, and therefore we need to play our role properly. And the biodiversity, I just borrowed this definition from UNESCO as the diversity of all living forms at different levels of complexity. It can be at gene level, species level, ecosystem, landscape, seascape. It's also economically important. It's a large part of the world's economy is dependent on biodiversity. In addition to that, our daily life in some countries, up to 80%, is completely dependent on biodiversity. Therefore, it's important to study biodiversity and have a better understanding about that. And the studies that have been done pointed out that there is a 58% decline in vertebrate populations during 42 years, which is a big number. More than 23,000 species are approaching extinction. Some reports saying that it is an underestimation. This number is even bigger than this. This not only threatens wildlife, but also global food supply. I mean, our life, our food supply. On the other hand, we can seek for new resources. It's like an opportunity. For example, FDA-approved drugs from marine species in preclinical and clinical phases. So, considering the importance of biodiversity, UN named last decade, I mean this decade, I should say, probably ending with COVID-19, which is not nice, but at least it was important attention, a lot of attention to biodiversity. And we set some goals, the global goals, which few of them are directly related to biodiversity. And in order to have enough and healthy food supply and water supply, it's important to have a better understanding of biodiversity and ecosystems. There have been an independent or parallel efforts contributing to this question, trying to address this question. And one of the earlier one is probably in animals is 29 Mammals project started in Sweden by Kerstin Lindblad's group. That's my former department. The same for 200 mammals, 10 kb, bats 1k, VGP also, which is really important, and etc. But probably the most recent and important one, and maybe the most ambitious project, is Earth Biogenome Project, which is a moonshot of biology. That aims to sequence, catalog, and characterize the genome of all of the Earth's eukaryotic species over a period of 10 years. So it's like a marriage of the technology and biology, application of sequencing methods, computational methods of computational biology, machine learning, and AI. So it's really it is really a big project. And within this project and projects alike, we have been using different sequencing technologies like Illumina, that you're all familiar with that. And in the beginning of this decade, we had a new invention of long sequencing, which some people call it third generation sequencing. One of the big players is nanopore sequencing technology, as well as Pacific Bioscience company, which use smart cell sequencing to generate long read. In addition to that, the combination of different techniques, we can develop new methods. For example, HiC that you can see in this figure. So the idea or application of HiC is to study the chromatin contact or chromatin interaction. But we can also use the same technique in order to create contiguous, scaffold contiguous sequences. Last but not least, 10x Genomics, which used to provide service for whole genome sequencing and its application in genome assembly. But they stopped this service maybe a year ago. Sorry. So with this big data, one of the things that we need is strong computational resources, which you see in this picture. And in Sweden, we have UPMAX, which is one of the largest one in the region. Sorry. So what we gain by studying biodiversity, I just list few of the important applications, and then after one slide I will give you 3 examples. And I start by saying that with this we can have a better understanding on different evolutionary processes. We can also monitor subpopulations over time and have better management. Also, we can learn a bit more about the fate of endangered species, which some people categorize as conservation genomics, and Stefan is going to talk about that in the next presentation, I guess. As well as this, we can learn about the reasons of extinction, also the importance of a species to the functioning of ecosystems. Also, it provides opportunity to learn about the possibility of restoring ecosystems critical to human survival. Last but not least, probably one of the main applications will be enhanced control of pandemics. We are in omics era from 2000, after human genome, the first draft human genome, we could see the birth of genomics application in many different studies. And here, this figure is I just generated it searching PubMed for different omics terms. And genomics, as is expected, is one of the earlier methods used to study biodiversity. So today, most of the examples that I present is from genomics. So yesterday we heard a lot about the application of genomics in domestic animals, like genomic selection in livestock. You can also use it in aquaculture and other sectors. We can also use these techniques to study domestication events and also natural population. We can use this for comparative genomics. One of the interesting application of this technique can be studying environmental DNA, which I have one example for that, one or two. We also use this to have a better management when it comes to invasive species and biosurveillance. The first example is about domestication of rabbit. So domestication events on different species was a turning point in human history, and made us more dependent on these resources, and also provided an opportunity to form new breeds for different use. One of the special factors about rabbit is that the wild ancestor still exists, so we can make a direct comparison between the domestic groups or breeds with wild rabbits. In Iberian Peninsula, we collected wild rabbit samples from different regions, and also southern France, which is a region that the domestication of rabbit happened. And in this bar chart, we are showing the nucleotide diversity. As you can see, in the wild rabbits and southern France, we see much higher diversity compared to domestic breeds. The interesting thing is that it is quite polymorphic species. We have 50 million, we identify 50 million SNPs. And from these, only 20 SNPs were fixed in either groups. So the domestication signature that we saw was more like a shift in allele frequency rather than fixation. The selection signals that we identified were mostly located in non-coding conserved regions, and they were associated with genes showing high expression in brain and neural development, which suggests, and we also looked into these the impact of these genes on different phenotypes of mouse, and we could see a significant enrichment of behavioral traits. So, which kind of makes sense, because behavior is probably the main trait under selection when it comes to domestication. We extended this project and studied the morphological differences between domestic and wild in brain. And this is an MRI of the wild and domestic rabbit brain, and one of the things that we identified was morphological differences in amygdala, for example. We also complemented it with RNA-seq data, and we could see there is a significant differential expression of genes in amygdala and hippocampus, and showed and suggested that there is an enrichment of genes in dopamine signaling. So with this information, I mean, one of the application which was suggested was by Carl Gustav Tholin, that maybe it provides an opportunity to domesticate the rabbit. So it's a topic for itself. I don't have any intention to go into that, but it was just one example to show the application of omics data to understand the domestication events, and also extend it to more like mini phenotypes. So the next example is about Atlantic herring, which is one of the main projects in Leif's group. And I think Leif Anderson does not need any introduction. We are all familiar with his work in domestic animals and also in general animal genetics. So this project I started in his group as a master thesis. And the data that we had was pool-seq data from different populations, and the idea was to identify genetic differentiation between different pools that you can see in this map. In Baltic Sea, we have low salinity, and when you go to Kattegat and Skagerrak, we see pools living in higher salinity, and the Atlantic Ocean with highest salinity up to 35. So this was a new pipeline that I developed, and we called it exome assembly. Don't confuse it with exome sequencing; it is different, and I explained it here. So what I did, I generated a transcriptome assembly, and as you can see, this is like the coverage of the transcript of the RNA-seq, and the red one is the coverage when I align the genomic data. So you see a dip when it comes to potential exonic region. What I did, I aligned the genomic data, and then I extracted the orphan reads and unmapped reads, and did a mini assembly. So it was computationally very intensive. I think I did more than 35,000 assemblies for this part. The beauty of this technique is that we could go from transcript level, from exon level, into non-coding region of the gene, like intronic region or promoter region. So after using this as a template, we could align the genomic data of the pools of different populations, and then identify differentiation sites, which using those sites we can see a beautiful clustering or distinction between Baltic population and Atlantic groups. So what by just looking into exome assembly, we do see some differentiation, but it is quite a scatter because of the nature of the data that we had. But thanks to advances in sequencing technology, we could generate a high quality draft genome assembly, and you can see this signal that we see in exome assembly is even larger, and you see this beautiful extension of the signal across the even bigger region. So again, we could advance this in the even further by using PacBio data, nanopore sequencing technology, Illumina data, and Hi-C, as well as linkage maps. So it's probably one of the high quality chromosome level assembly of non-model species. Saying that, despite of having this really high quality assembly, there is still dark side of the genome. For example, the sex associated region or sex determination region of this chromosome of this species was very challenging to identify, and we published it 2 weeks ago, I think, in PNAs. And I suggest, if you are interested to learn about the dark side of the genome and how we can use unmapped reads or junk data that we usually don't look into, that please go and read this paper. The interesting thing is that it got many media attention as well. Now I switch and talk a bit about comparative genomics. In the beginning, as I told you, we have COVID-19 situation, and at the same time we have this beautiful data set that we can identify which of the other species may be affected. So COVID-19 or a coronavirus in this case is sort of entering into cells by using the ACE2 receptor. So the idea here was to see how many other species have this receptor. And in this project, they used 410 vertebrates, and they identified intermediate hosts. And most of them were, unfortunately, wildlife and endangered species. So it's very important this is one of the important application that identify these potential hosts. And if they are endangered species, then we can use this information for better management. And one of the application is in environmental DNA, which we can see that as a tool in conservation for monitoring past and present biodiversity. We can learn about ancient and modern environments. As an alternative to fish survey, for example, instead of net or electrical fishing, we can use this technique. So electrical fishing is that there is an electric shock in water, and then fish are paralyzed, and then we can check the species, but it means that we have to be at the right time and right place. So when it is not possible, we can use eDNA. This is one of the fun things that I did. I went to the river in Uppsala and I got a bucket of water, went to lab, extracted DNA, and I used universal primers, and I could amplify fish-specific fragments. Unfortunately, I didn't get the money to do the sequencing, but it is one of the applications. One of the other projects that I had in mind, but I was a bit late, it was 2017, which I came up with this idea, but a few weeks later there was a publication which shows the application of DNA metabarcoding to investigate honeybee foraging, with correlating it with floral availability in different regions. And here you can see the correlation of the identified sequences through DNA associated with the floral availability in different regions of this site. Last but not least is an example about invasive species and biosurveillance. Guess what? Yes, climate change can contribute to this to a large extent. We have negative ecological and economic impacts, and one of the drivers of biodiversity loss is climate change. Using genomic tools provide fresh insight to problems like this, and provide managers a more effective means to identify potential threats. We can use metabarcoding and other techniques to study population genomics like RADseq or GBS that you heard about, application of GBS in camel breeding yesterday. And the outcome will be prevention, for example, or better eradication strategies, or have a bit more effective targeted control to minimize hybridization. This is quite extensive topic that we can talk hours, which is not my intention. So to wrap up, biodiversity matters. The clock is ticking, because if we lose, if we break the chain, the ecosystem will be disrupted, and it will have sometimes irreversible impact on ecological interactions. There are some critics about this phylogenetic tree, but the point here is that the human activity has much higher speed than evolution. So evolution does not have enough time to sort of regrow and diversify the available species. So this is the figure that you have seen. It's boring. I'm not going to bore you, but I try to say a joke about this, because the first time that I saw this, it was 2008 and 9. And I used to say, if it goes like this, we will get paid if we send our samples to sequencing centers. But we all know that's not the case. But thanks to this marriage of the biology and technology, we have this possibility to take labs to nature. So now we can use, for example, nanopore sequencing to study species at sites. Another application is handled real-time PCR, that we can grab this, go to forests and study invasive species. So the examples that I gave you is most of them is part of the time that I spent in Leif Andersson's group. And Leif is here. I'm here. It's his team. It's probably a bit old picture, but it's me when I was a bit younger. And I would like to thank SciLife Lab, which is a great facility providing this opportunity to do world-class studies. Also Max the server and my team at NBIS. With this, I will be happy to take any question, and I hope this will be an introduction to upcoming lectures or talks that I'm looking forward to hear. Thanks for your attention.